The red archived the Perseverance rover landed in Jezero crater on February 18, 2021 at 20:55 UTC. The landing was precise to within a few kilometers of the target zone, guided by the Terrain Relative Navigation System that compared real-time camera images of the Martian surface to preloaded orbital maps and made autonomous corrections at 200 meters per second. The rover carries 7 scientific instruments, 23 cameras, 2 microphones, and a sample caching system capable of collecting and hermetically sealing rock cores for eventual return to Earth. It is the most sophisticated robotic explorer ever sent to another planet. Its primary science mission is to search for signs of ancient microbial life in what 4 billion years ago was a lake fed by river delta deposits, a body of standing water that persisted for potentially millions of years under conditions that models suggest were compatible with the chemistry of life as we understand it. The numbers that motivated this mission are straightforward. Mars 4 billion years ago had liquid water on its surface, a denser atmosphere, a magnetic field partially shielding it from solar radiation, and the full suite of elements required for carbon-based biochemistry. If the origin of life is a natural consequence of chemistry operating in the right conditions over sufficient time, and if the right conditions existed on Mars for millions of years, then the probability of life having arisen there is not negligible. It is, depending on which model of abiogenesis you accept, potentially quite high. The Perseverance mission was designed to find evidence of what happened. What it found has been reported in the scientific literature with appropriate precision and appropriate caution. What the accumulated data implies, assembled into a coherent picture, has not been stated in any press release or any public communication from any space agency. This is where the terror begins. The first discovery that should have received more attention than it did involves the organic molecules detected in Jezero crater by the Sherlock instrument, a deep ultraviolet Raman spectrometer that can identify organic compounds in rock samples without physical contact. Sherlock detected organic molecules in multiple rock samples in Jezero crater, including in the Wildcat Ridge sample collected in July 2022 from what appears to be a mudstone formed in an ancient lake environment. The detection of organic molecules on Mars is not itself surprising. Organic molecules are delivered to planetary surfaces by meteorites and can be produced by non-biological chemical processes. But the specific distribution of organic molecules in the Wildcat Ridge sample is not what would be expected from purely abiotic delivery or synthesis. The organics are concentrated in specific layers and are associated with sulfate, minerals in a pattern that on Earth would be interpreted as biosignature evidence, the chemical signature of biological activity. The paper reporting this detection, published in Science in July 2023, was careful to note that the organic molecules could have abiotic origins. What the paper did not state in its abstract, though it acknowledged in its discussion section, is that the spatial distribution and mineralogical association of the organics is more consistent with biological concentration than with any known abiotic process studied at Jezero crater. The number of abiotic explanations that have been proposed and found consistent with the specific pattern observed is 0. The number of biological explanations that are consistent with the pattern is more than 1. This does not prove biology, but it is not the distribution of noise. It is a signal that does not have a non-biological explanation yet identified. The second discovery involves the subsurface radar data from the RIMFAX instrument, a ground-penetrating radar that has been operating continuously since landing and has mapped the subsurface structure of Jezero crater along every traverse the rover has driven. The RIMFAX data, published in a series of papers in 2023 and 2024, reveals a subsurface stratigraphy that is more complex than the pre-mission model's predicted dent that contains evidence of multiple episodes of water table variation over geological time. Specifically, the data shows evidence of ancient groundwater upwelling into the crater floor sediments, a process that on Earth is associated with hydrothermal systems, which are among the most productive environments for microbial life on our planet. The hydrothermal interpretation of the RIMFAX data is consistent with the mineralogy of the surface samples, which include minerals that form in the presence of hot water rich in dissolved ions. Hydrothermal systems on Earth do not merely support life. They may have been the environment in which life on Earth originated. The discovery that Jezero crater had an ancient hydrothermal system is not definitive evidence of life. It is evidence that Mars had the most productive known environment for the origin of life, operating for a duration sufficient, by any model of abiogenesis we currently possess, for life to have arisen. This finding did not make the front page of any major newspaper. It was reported accurately in the scientific literature and briefly noted in a small number of science journalism outlets. The implication of the finding, that Mars was not merely habitable, but was arguably optimally configured for the origin of life during its early history, was not stated by any official source. The third discovery involves the methane detections by the Curiosity rover in Gale Crater, which have been ongoing since 2014, but whose full implications have only recently been appreciated. Methane in the Martian atmosphere is significant because it has a photochemical lifetime of approximately 300 years. Any methane present in the Martian atmosphere must have been produced recently, either by geological processes such as serpentinization, the reaction of water with iron and magnesium silicates, or by biological processes. Curiosity has detected methane in background concentrations of approximately half a part per billion by volume, with periodic spikes reaching up to 21 parts per billion, observed repeatedly and most prominently in the Martian summer when temperatures are highest. The seasonal variation of the methane signal is consistent with biological methanogenesis, the production of methane by microorganisms, releasing gas stored in subsurface ice during warmer temperatures. The seasonal variation is also consistent with geological serpentinization with a similar temperature dependence. The ESA Trace Gas Orbiter, which entered Mars orbit in 2018 specifically to map trace gases including methane, reported in 2019 that it had found no methane. This contradicts the Curiosity surface measurements. The contradiction has not been resolved. The two instruments cannot both be correct simultaneously, and the resolution of the discrepancy has significant implications for whether the methane is real, widespread, or locally produced. The absence of a clear resolution of this contradiction from any official source, despite 5 years of continued investigation, is itself a data point. Either the methane is real and local, or the surface measurements are incorrect, or the orbital measurements are missing a real but spatially constrained signal. None of these explanations is comfortable, and none has been publicly endorsed by either agency. The fourth discovery involves the ancient magnetic field record preserved in Martian rocks, and what the latest analysis of this record implies about the timeline of Mars's habitability. Mars does not currently have a global magnetic field. Its dynamo shut down approximately 4 billion years ago, after which the solar wind began stripping the Martian atmosphere. This loss of atmospheric pressure is the primary reason Mars is now a cold, dry, irradiated desert rather than the warmer, wetter world it once was. The timing of the dynamo shutdown has been studied using orbital magnetic measurements and, more recently, using in-situ measurements from the InSight lander's magnetometer. The InSight data, published in 2022, revealed that the ancient magnetic field at InSight's landing site in Elysium Planitia was significantly stronger than orbital measurements had suggested, and that the field was still active as recently as 3.9 billion years before present. This is the same age as the oldest known evidence of life on Earth. If the magnetic field on Mars persisted to 3.9 billion years ago, then Mars was magnetically shielded from solar radiation during exactly the period when life on Earth was originating. The overlap in timing is precise. It is not a coincidence in any trivial sense. It means that the conditions on Mars at the time when life is thought to have arisen on Earth, meaning liquid water, magnetic shielding, atmospheric pressure, and the right chemical environment, were present simultaneously on Mars as well. The probability that life arose on Earth during this period, but not on Mars, given that all the relevant conditions were present on both planets simultaneously, cannot be calculated with precision. But any honest calculation produces a number that is uncomfortable. The fifth discovery involves the ancient river delta in Jezero crater, which the Perseverance rover has been exploring since April 2022, and which has revealed a degree of geological complexity and a diversity of mineral types that exceeded all pre-mission predictions. The delta is a fan-shaped deposit of sedimentary rock formed by an ancient river that emptied into the lake that once filled Jezero crater. River deltas are among the most productive environments for life on Earth because they concentrate organic material and provide a gradient of chemical conditions from the river water to the lake water that supports diverse metabolic strategies. The Jezero Delta contains sandstones, mudstones, sulfate minerals, perchlorate minerals, and organic molecules in a stratigraphy that indicates multiple episodes of deposition under changing environmental conditions spanning potentially millions of years. The diversity and complexity of the delta sediments is, on Earth, associated with rich biological ecosystems. The absence of confirmed biosignatures in the delta samples analyzed so far by Sherlock and the other contact science instruments is the central null result of the Perseverance mission to date. But the null result is not a negative result. The instruments aboard Perseverance were not designed to definitively detect or rule out life. They were designed to identify samples that should be returned to Earth for analysis by laboratory instruments orders of magnitude more sensitive. The samples Perseverance has collected and sealed are the most carefully chosen geological samples from another planet ever assembled. What they contain will not be known until they are analyzed on Earth, which will not happen until the Mars sample return mission is completed, currently projected for the mid-2030s at the earliest. The sixth discovery involves the perchlorates in the Martian soil and their implications for both the habitability of Mars and the interpretation of the Viking lander results from 1976. The Viking landers conducted a labeled release experiment in which Martian soil was mixed with a nutrient solution containing carbon-14 labeled organic compounds. The experiment produced a positive result. Carbon-14 labeled gas was released from the Martian soil, suggesting metabolic activity. The result was subsequently attributed to the presence of highly oxidizing chemicals in the Martian soil, initially hydrogen peroxide and later perchlorate, which could chemically oxidize the organic compounds without biological involvement. The perchlorate explanation of the Viking results has been widely accepted for decades, but the perchlorate concentrations measured by Curiosity and Perseverance are sufficient not only to explain the Viking results chemically, but also to provide an energy source for microbial metabolism. Perchlorate-reducing bacteria are a well-studied metabolic group on Earth, capable of using perchlorate as a terminal electron acceptor in the absence of oxygen. The perchlorate in Martian soil, which was previously seen only as an obstacle to life, is also a potential fuel source for a specific type of microbial metabolism that does not require sunlight, oxygen, or surface water. Subsurface microbial communities in the deep subsurface of Mars, protected from radiation and accessing liquid water from brines or ice, could in principle sustain a perchlorate-based metabolism for geologically significant timescales. This has been noted in the scientific literature. It has not been communicated as a priority finding by any space agency. The seventh discovery involves the recurring slope lineae, dark streaks that appear on Martian slopes during warm seasons and fade during cold seasons, first definitively identified in 2011 from Mars Reconnaissance Orbiter images. The recurring slope lineae were initially interpreted as possible evidence of liquid water flowing on the present Martian surface, a finding that would have enormous implications for present-day habitability. Subsequent analysis, including spectroscopic measurements showing the absence of hydrated minerals along the lineae, has suggested alternative explanations, including dry granular flows and dust avalanches. The liquid water interpretation has been largely abandoned by the mainstream science community, but the controversy is not fully resolved. The absence of hydrated mineral signatures in orbital spectroscopy does not rule out transient liquid water. That evaporates before hydrated minerals can form and be detected at the resolution of the orbital instruments. The recurring nature of the lineae, their correlation with temperature, and their seasonal behavior remain consistent with a liquid briny water explanation, and the alternative explanations have not produced a quantitatively satisfying model of the observed behavior. This is a finding whose implications, if the water interpretation is correct, are profound. It has been managed rather than resolved. The eighth discovery involves the subsurface radar detections by the MARSIS instrument on the Mars Express orbiter, which in 2018 reported a bright radar reflection approximately 1.5 kilometers below the south polar ice cap, consistent with a subglacial lake of liquid water. Subsequent analysis identified additional bright reflections in the same region, consistent with a system of subglacial lakes similar to the subglacial lake systems beneath the Antarctic ice sheet on Earth. The Antarctic subglacial lakes are inhabited by microbial communities that have been isolated from the surface for millions of years. If the Martian South Polar bright reflections are subglacial lakes, they represent the most plausible current habitat for life on Mars. The interpretation has been challenged by some researchers who argue that the bright reflections could be caused by highly conductive minerals rather than liquid water. The debate continues in the scientific literature. The Mars Express mission continues to collect data. No consensus has emerged. The possibility of liquid water lakes currently existing beneath the Martian South Polar Cap, potentially inhabited by the descendants of whatever life existed on early Mars, is a possibility that has not been publicly communicated as a priority finding, despite being supported by orbital radar data from a scientific instrument specifically designed for subsurface sounding. The ninth discovery is the one that assembles all the others into a picture whose implications extend beyond Mars science into the deepest question in all of biology and all of physics. In 2022, a research team analyzing the Perseverance rover's first year of surface operations published a comprehensive assessment of the habitability of Jezero crater across geological time. Their conclusion, stated carefully in the language of scientific literature, was that Jezero crater satisfied every criterion for habitability that has ever been identified in the study of extreme environments on Earth, across a time period of at least 1 million years and potentially up to several hundred million years during the Nokian and early Hesperian epochs. Every criterion, not most criteria, not several criteria. Every criterion that the scientific community has identified as necessary and sufficient for microbial life, based on the complete inventory of life on Earth, was simultaneously present in Jezero crater for a duration that exceeds the estimated time required for life to originate by any current model of abiogenesis by a factor of between 10 and 100. The paper that contains this conclusion was published in a peer-reviewed journal. It was not reported in a single major news outlet. It was not the subject of a NASA press conference. Its implications, stated plainly, are that if life does not arise under the conditions that existed in Jezero crater for the duration those conditions persisted, then life does not arise under any conditions we have ever identified, on Earth or anywhere else, and our entire theoretical framework for understanding the origin of life is wrong in a fundamental way. And if that framework is wrong, then the expectation that life should be common in the universe, which is the foundation of every optimistic answer to the Fermi paradox, is also wrong. And if that expectation is wrong, then the silence of the cosmos is not a puzzle. It is the answer. Mars was the test. Mars had every condition required. Mars is now dead. And the question of whether Earth is an exception or whether life on Earth is itself something we do not fully understand is the question that the 9 discoveries assembled here are forcing us to ask, whether we are prepared to or not. We are not special. We may be wrong. And the archive in the red rock, sealed in sample tubes on the floor of an ancient lake, is waiting to tell us which one it is, if we can get it back before we decide we already know the answer.